welcome to All Things Pharmacy. Today we're going to be talking about the mechanisms of drug absorption, the three main ways in which the drug is absorbed into the body, from the GI lumen into the tissues surrounding the lumen. And this is through transcellular or intracellular transport. Second one being paracellular or intercellular transport. And the third one being vesicular transport. I'm just going to give a brief overview of those three mechanisms of transport. And for transcellular, it means that the drug goes through the membrane. So between the GI lumen and the tissue, there's a membrane that separates them from being in direct contact with each other. And so when a drug is absorbed transcellularly, it passes through the membrane, which is the cells of the membrane. And transcellular absorption can be further divided into different types. For example, there's passive diffusion, and there's also carrier-mediated transport, which we will further discuss. So transcellular transport is through the actual cells. And then there's paracellular transport, which is in between the cells. So the drug will pass in between two cells, and which is through like a junction, because they also have cell junctions, and it will pass the junction and go directly into the tissue. It goes directly into the capillary or whatever blood supply is there. It goes into the systemic circulation. The third one is vesicular. As you can tell from the word, there's vesicles involved, and so from the lumen, the drug is taken in through vesicle. A vesicle takes the drug from the extracellular into intracellular, and then the vesicle itself with the drug inside, it travels through the cell, and then the vesicle is released on the other side of the cell through a process called endocytosis. From that, it goes into the capillary or whatever blood supply, either a vein or artery. Now we're going to focus on transcellular transport, and it is divided into four, which is passive diffusion, pore transport, ion pair transport, and carrier-mediated transport. Now first we're going to discuss passive diffusion. So in passive diffusion, the drug will move from a high concentration to a lower concentration. Remember that when you ingest the drug will be in the GI lumen. So this is before it gets absorbed into the capillaries. And so because of that, there will be a higher concentration of the drug in the lumen than in the capillary. So the drug will move from a high concentration to a lower concentration. This is also called the sink condition. It's called the sink condition because as soon as the drug gets into the capillary, into the systemic circulation, it flows away along with the blood. And so which means that in the capillary, at the lumen, there will always be a lower concentration of the drug because it will be taken away as the blood flows. And so this sink condition will always be there. The Fick's first law of diffusion is used to best express this passive diffusion, and this law states that drug molecules diffuse from region of higher concentration to a region of a lower one until equilibrium is attained. And also the diffusion is directly proportional to the concentration gradient across the membrane. There's a mathematical equation that represents the Fick's law. Another type of transcellular transport is pore transport, and this involves water-soluble drugs. The water-soluble drugs will pass through the membrane using aqueous channels, and the osmotic differences is what causes the drug to move from the lumen to the capillary. The hydrostatic pressure or osmotic difference is the driving force which causes the drug molecules to move. Besides being water soluble, for drugs to use the poor transport mechanism, they also have to have low molecular weight. The rate at which the water soluble drug moves depends on the number of pores and the sizes. Next, we're going to be talking about the ion pair transport, and this is for the movement of ions through the membrane from the lumen to the capillary. And an example of a drug that uses this type of transport is the quaternary ammonium compounds and the sulfonic acid. These drugs mainly remain ionized under all pH conditions, and when a drug is unionized, which means it's more lipophilic, which it means that it's able to cross through the membrane more easily than drugs which are ionized. So drugs which are ionized have to find a special way of transport so that they can cross the membrane, because they are not lipophilic, and so this is why the ion pair transport is there in the body to facilitate this movement. The ion pair transport, as you can see, there's the word pair, which means that the ionized drug will pair with another endogenous anion in the body, and it will form a complex with this endogenous anion, and this complex is what crosses through the membrane. When it gets to the other side of the membrane in the capillary, the complex dissociates and it separates. So now in the blood there will be a free drug and an endogenous anion, which is the same thing found in the lumen. However, they had to cross together through the membrane. An example of a drug that has this type of transport is propranolol. Next, we're going to be talking about carrier-mediated transport, and this transport is for polar drugs. The drug moves depending on the concentration gradient, the electrochemical concentration gradient, and also the partition coefficient. The carrier-mediated transport involves a carrier, and the carrier forms a complex again with the drug molecule, and it crosses the membrane. However, this time when it gets to the other side, the carrier leaves the drug molecule in the blood, in the capillary, and it goes back to the other side of the cell, which is to the lumen, to get a new drug molecule so that it can also cross, and just like that the process will continue. There are two types of carrier-mediated transport, which is facilitated diffusion and active transport. Facilitated diffusion involves the drug molecules going down the concentration gradient, which means from a high concentration to a low concentration, and this is faster than passive diffusion. If you remember, we said that passive diffusion, it also goes down the concentration gradient, and a difference between passive diffusion and facilitated is that facilitated has an intrinsic factor, which is another molecule which the drug will bind to, and then after binding to that, it will then go on to bind to the carrier now. So without this intrinsic factor, the drug cannot bind to the carrier by itself. When the complex of the drug molecule, intrinsic factor, and the carrier gets to the other side, which is in the blood, the complex now dissociates, and they'll be left with the drug and the intrinsic factor separately. They're not bound to each other, and then the carrier will remain in the membrane, and it will go back to the lumen and start the whole process again. It will bind to an intrinsic factor, and to that intrinsic factor they would have already been bound to a drug molecule. The intrinsic factor is a glycoprotein that is produced by the gastric parietal cells. Another type of a carrier mediated transport is active transport, and this happens against the concentration gradient, so the molecules will move from low concentration to a high concentration, and this involves the use of carrier proteins as well. And so now, just to recap on what we've just learned, we've learned that there's mechanism of drug absorption, and these are transcellular, intercellular, and vesicular. And there's also transcellular can be further divided into passive diffusion, pore transport, ion pair, and carrier mediated transport. Passive diffusion has a driving force, which is concentration gradient. It has a sink condition, it has fixed law. Pore transport, there's osmotic differences, and this is for low molecular weight water soluble drugs, and they go through aqueous filled channels. And then there's ion pair, which is for ionized drugs under all pH conditions, and they form a complex with a carrier, so this is where carrier is used. And then there's a carrier mediated transport, and with this, not only is there a carrier, but there's also an intrinsic factor, and there's also a concentration gradient, and the partition coefficient plays a role in the movement of drug molecule. And lastly, the carrier mediated transport can be further divided into active transport and facilitated diffusion. So this is the end of the lesson. Thank you for watching. Don't forget to subscribe, to like, to comment, and to share with your friends. Until next time.